Hello friends, welcome to my channel Easy Life Science. Here I am talking about the questions which will be asked for a set exam. So please subscribe my channel Easy Life Science, like my video, share it to your friends and don't forget to press the bell icon so you will get the notification of newly uploaded video. Here starting from the question 15. The question is asked, match the disease with causative organism and a affected crop. One table is given. In first column disease, in second column causative organism, and in third column crops. The first, that is a P, powdery mildew. The powdery mildew is caused by the Yersinia auranti. This organism causes a powdery mildew and this is a disease of a tomato plant. So P match to the 3 and in a crop it match to a second. P3 second. Then disease, that is a white rust. The causative organism for a white rust is a albigo candida. It is a one fungi and it is a disease of a mustard. So Q1 and 4th. Then R, downy mildew. The causative organism for downy mildew is Personscleropora philippinensis and it is a disease of a corn. The late blight disease. Causative organism for this disease is a Phytophthora impestans and it is a disease of a potato. So the option B is the answer of this question. Then moving to the next question, match the pairs group 1 and group 2 or group 2. In group 1, phospholipase C, inositol triphosphate, diacylglycerol and phosphatidic acid. The phospholipase 3 is a class of a membrane associated enzyme that cleaves phospholipids. The phospholipase C plays a role in a signal transduction. This enzyme cleaves a phosphatidyl inositol, that is a PIP2, into diacylglycerol, that is a DAG, and a inositol 1,4,5 triphosphate, that is a IP3. Overall, the PIP2 get converted into a DAG and a IP3 through the action of a PLC, that is a phospholipase C. So from group 2, the third point, that is a hydrolyzes a phosphatidyl inositol triphosphate or a bisphosphate, it matching to a phospholipase C. Then a Q, inositol triphosphate. The inositol triphosphate, it stimulates the release of a calcium from a intracellular stores, that is a endoplasmic reticulum. The inositol bisphosphate or a triphosphate, it serves as a secondary messenger. Activator of each PLC is a variable, but typically it includes a heterotrimeric G protein. Sometimes it may be a protein tyrosine kinase or a small G protein. Calcium ions or phospholipids are the activators of phospholipase C. The phospholipase C hydrolyzes the phosphatidyl inositol triphosphate to a secondary messenger inositol 1,4,5 triphosphate, that is a IP3 and a DAG. The IP3, that is a inositol triphosphate, it induces a transient increase in intracellular precalcium, while the DAG, that is a diacylglycerol, activates a protein kinase C. So for inositol triphosphate, The group second, the first point, it stimulates a release of a calcium from intracellular stores, that is from endoplasmic reticulum. Then the from group 1, diacylglycerol or a DAG. The DAG get phosphorylated to a phosphatidic acid, and this diacylglycerol activates the protein kinase C. Then the yes from a group first, that is a phosphatidic acid. The phosphatidic acid are anionic phospholipids important to a cell signaling and direct activation of lipid gated ion channels. Hydrolysis of phosphatidic acid gives rise to a one molecule of glycerol, one molecule of phosphoric acid, and a two molecule of fatty acids. The phosphatidic acid contributes about 0.25% of phospholipids in a bilayer. So for yes from group 1, it matches to a second point in a group 2, stimulate ion channels or activates various enzymes. So for question number 16, the option D is a correct option. Then the next question, which one of the following methods is incorrect with respect to a haploid plant production? It can be produced. The haploid plant production can be done by using an unfertilized egg cell. It can be done by a isolated pollen culture or by a distant hybridization followed by a selective elimination of a chromosome of one parent. But it cannot be arise from a new cellular tissue. So the B option is the answer for this question. Then moving to the next question. Chloramphenicol and tetracycline are a broad spectrum antibiotic which inhibit bacterial growth by targeting. Here in this question, the target site for chloramphenicol and tetracycline are asked. These two antibiotics are a protein synthesis inhibiting antibiotic. They inhibit the protein synthesis of the bacteria or a pathogen. So this bacteria does not grow due to the application of chloramphenicol and tetracycline. The chloramphenicol, it inhibits microbial protein synthesis by binding to a 50S subunit of a 70S ribosome and inhibiting the action of peptidyl transferase. Thus, it prevents peptide bond formation. This mechanism also prevents the binding of amino acyl tRNA to the peptidyl transferase active site. So this is the activity of a chloramphenicol on a target cell, whereas the tetracycline, it binds to a 30S ribosomal subunit, hindering the binding to the amino acyl tRNA to the acceptor site on the mRNA ribosome complex. So the option B is the answer for this question. The other option like cell wall synthesis, RNA synthesis and a DNA synthesis. The antibiotic like phosphomycin, cycloserine and beta lactams are a cell wall synthesis inhibitors. This is the activity of a tetracycline. The tetracycline binds with a 30S subunit of a ribosome. It hinders a binding of amino acyl tRNA to the A site, that is the acceptor site, and hence it blocks a further protein synthesis, and due to no protein formation, the target organism get killed. This is the activity of a tetracycline. Here in this image, a mechanism of antibiotic action is given in a typical cell. Some antibiotic can inhibit a protein synthesis by binding to a ribosomes. Some can inhibit a nucleic acid synthesis, some inhibit the cell wall synthesis, some can disturb the cell membrane function, and some can block the pathway which are useful for metabolism. Here in this image, some antibodies are given and their mode of action is given. The cell wall synthesis inhibitor, for example, beta lactams, glycopeptides, phosphomycins, bacitracin, alphosphelin, these are the cell wall synthesis inhibitors. Then DNA gyrase inhibitors, quinolones and comeromycin are the DNA gyrase inhibitors. The RNA polymerase inhibitor, that is rifampicin, RNA synthesis inhibitor, that is ansamycin, the protein synthesis inhibitor binding to a 30S and a 50S subunit tetracycline aminoglycosides, macrolides, these are the protein synthesis inhibitor, folate synthesis inhibitor, that is sulfonamides, cell membrane synthesis disturbers, lipopeptides. This is about the mechanism of action of some antibody. Here in this image, same image is there. The antibiotic with their target site, cell wall synthesis inhibitor, DNA synthesis inhibitor, metabolic pathways inhibitor, ribosome binders, and plasma membrane disturbers. You can take a screenshot of it. Then moving to the next question. Which of the following radiation method is used in preserving a food from spoilage by a microorganism? The beta rays are used for the food preservation. So these are ionizing rays. So option D is the answer of this question. The next question, which part of a lipopolysaccharide has a toxic property that make some bacterial infection a potentially serious medical problem? The gram-negative bacterial cell wall is made up of two layers. The lipopolysaccharides are present outer layer, and this LPS or lipopolysaccharides are termed as endotoxin. The LPS are a large molecule consisting of a lipid and a polysaccharide, and these are bacterial toxins. They are composed of O antigen. So, lipid A portion is having a serious medical problem. So, option B is the answer of this question. Then next question, microbes make up about dash of Earth's living material by a weight. Option B is the answer of it. 2/3 part of Earth living material is of microbes. Then moving to the next question, a highly contagious viral disease, measles, is caused by. Here the question is asked about a measles virus and its genetic material. The genetic material of a measles virus is a single stranded RNA. Then next question, secondary metabolites such as penicillin from a mold are produced during dash dash. Here about the growth phase, the secondary metabolites are formed in a idiophase. Idiophase, the phase in a growth of a culture during which a secondary metabolites are produced. Compounds produced in the idiophase have no direct relationship to the synthesis of cell material and a normal growth of the organism. So growth phase is also called as a trophophase, and in a trophophase primary metabolites are produced, but in a production phase or in a idiophase there is a production of secondary metabolites. The penicillin is the secondary metabolite and it is produced by organism Penicillium notatum. So option B is the correct answer for this question. In a lag phase, there is no any growth. Log phase, there is a cell division, and in a decline phase, the death rate is more than that of the growth rate. So, for this question, B is the answer. Then next, Dick test is used to assess the susceptibility to. The Dick test method of determining a susceptibility to the scarlet fever by a injection into a skin of zero point 0.1 cubic centimeter of scarlet fever toxin, a reddening of a skin in an area over 10 millimeter in a diameter within a 24 hours indicates a lack of immunity to the scarlet fever. So option B is the answer of this question. Dick test is used to assess the susceptibility to the scarlet fever. Then the next question, which of the following is a symbiotic bacterial genus that converts nitrogen into a nitrogen containing compound that are utilized by a legume plant to synthesize a amino acid? Here a question is asked on a symbiotic nitrogen fixation, a symbiotic association in between a legumes of a dicotyledonous plant and a nitrogen fixing bacteria. The rhizobium is a bacteria which have a symbiotic association to the legumes of a dicotyledonous plant. Here the rhizobium fixes a nitrogen. The nitrogen is made for a growth of a plant, whereas the rhizobium gets a shelter from a plant. So both the organism or both the partners in this association get benefited. So it is the symbiotic association. The other options, spirillum, azotobacter and clostridium, these are also example of a nitrogen fixing organism, but they don't have a symbiotic association with their partner. So option C is the answer of this question. Then the next question, what is the enzyme involved in a following reaction? One reaction is there, triglyceride in presence of three molecules of water get converted into a glycerol and a fatty acid. Which enzyme carry out this reaction? Options are there, glycerol phosphate dehydrogenase, glycerol kinase, lipase and zymase. The answer of it is a lipase. The lipase enzyme breaks down a triglyceride in presence of a water molecule into a glycerol and a fatty acids. The option A, that is a glycerol phosphate dehydrogenase. Glycerol phosphate dehydrogenase, this enzyme catalyzes the reversible redox conversion of dihydroxyacetone phosphate to a glycerol 3 phosphate, whereas the glycerol kinase, this glycerol kinase enzyme is encoded by a gene, that is a GK gene. It is a phosphotransferase enzyme involved in a triglycerides and a glycerophospholipid synthesis. The enzyme glycerol kinase catalyzes the first step of glycerol metabolism and transferring the triol into a glycerol 3 phosphate. Zymase, this enzyme convert a pro enzyme into a enzyme. So option C is the answer of this question. Then next question, keep this question. Folic acid synthesis in a bacteria is competitively inhibited by a sulfonamides. Which of the following essential component is replaced by a sulfonamides during the process of folic acid synthesis? The option C, that is a PABA, para amino benzoic acid. The para amino benzoic acid or a PABA, it is a precursor for a formation of folic acid through this reaction, and the folic acid is a important component for a synthesis of a DNA, RNA and a protein. The para-aminobenzoic acid, it is a substrate for the enzyme dihydropterate synthesis. But the enzyme dihydropterate synthesis, this enzyme get competitively inhibited by sulfonamide drugs. So this drug or sulfonamides blocks the active site of dihydropterate synthesis, and hence rather than a binding of PABA, the sulfonamide gets bind to a dihydropterate synthesis and reaction get blocked, and further there is no formation of the folic acid. So the sulfonamides are the competitive inhibitor of a reaction or an enzyme which play an important role in a folic acid synthesis. The next question: which of the following cellular structure are absent in eukaryotes and present at least in some prokaryotic organism? Here question is asked: absent in eukaryotes and present at least in some prokaryotes. Respiratory enzymes. The respiratory enzymes are present in a mitochondria of eukaryotic cell, and sometimes they are present in a peroxisome. So this is not an option. A ribosomes are present in a eukaryotes as well as a prokaryotes. So this is not an option. Nucleoid. The nucleoid only present in a prokaryotes, whereas it is absent in a eukaryotes. Third, and internal membranes. The internal membranes are present in eukaryotes. For example, nuclear membrane, endoplasmic reticulum, Golgi bodies. These are the internal membranes. Pili. Pili are absent in eukaryotes and present at least in some prokaryotes. So third and fifth, such combination is in option A. So option A is the answer of this question. Then moving to the next question. Granules are inclusion bodies found in a bacteria. Which of the following statements are true about the granules? Granules are a storage vessel for a carbohydrate and lipid. Is it true? Some granules supply a substances for a variety of metabolic processes. It is also true. Option third, or point number third, granules are dissolved in a cytoplasm. So it is false. The granules are not dissolved in a cytoplasm. Granules are membrane enclosed structure. It is also false. So point first and point second are true regarding to this question. Such combination is option C. So option C is the answer of this question. Moving to the next question. Match the culture media with their uses. In one column there are the medias, and in a right column there are the uses of that media. First, a complex media. The complex media are chemically undefined media for a growth of bacteria. For example, the nutrient agar and a macrocyst agar, the complex media, or a semi-synthetic media. They are chemically undefined media. Then second, that is a selective media. Macrocyst agar is a selective for the member of a family Enterobacteriaceae. As it contains a sodium taroclate, it favors the growth of a member of family Enterobacteriaceae. So such media are favors or used for a growth of selective organisms. For identification of a certain bacteria from a mixed culture, for identification of E. coli from a mixed culture like sewage, we can make a use of a macrocyst agar. So it is a use of a selective media. Differential media. The differential media can differentiate two groups of organism or two types of organism. For example, the McConkey's agar is also a differential media. It can differentiate coliforms and non-coliforms. The coliform organisms shows pink colored colonies on a McConkey's agar, and non-coliform organisms shows a yellow or a colorless colony on a McConkey's agar. As coliforms are lactose fermenting, the color of colony of them is pink, whereas the non-coliform does not ferment a lactose sugar present in a medium, so color of colony of this organism is yellow or a colorless. So we can differentiate the coliforms and non-coliforms by using such kind of differential media. Then enriched media. The enriched media are used for cultivation of fastidious organisms. The fastidious organisms are the organisms which require the extra growth factor for their growth. For example, the blood agar or a blood broth is an enriched media for pathogens of a human being. So, such combination is in option B. Option B is the answer for this question. Then the next question, among the following characteristics listed below, select those that are most appropriate for archaeobacteria. Archaeobacteria are known as ancient bacteria. These are extreme phytic organisms. They can live better at extreme conditions of environment. The four groups of archaeobacteria are there: methanogens, wall-less archaeobacteria, sulfate reducers, and thermophilic archaeobacteria, as well as halophilic archaeobacteria are there, and they are different from that of the eubacteria. The eubacteria or their cell membrane is made up of long chain hydrocarbons, and these long chain hydrocarbons are bounded to a glycerol by a ester linkage. But in case of archaeobacteria, there is a presence of long chain branched alcohols or phytanols, which are bound to a glycerol by a ether linkage, and this is a difference in between cell membrane of eubacteria and a cell membrane of archaeobacteria. Also, the translation process of archaeobacteria is sensitive to the action of a diphtheria toxin. So, point second and point fourth are true regarding to the archaeobacteria. The point third, first amino acid in protein synthesis is N-formyl methionine. This is wrong. This is true for eubacteria, but in case of archaeobacteria, the first amino acid at time of a protein synthesis is a methionine. So, point number second and fourth are true regarding to the archaeobacteria. Such combination is in option C. So the answer is option C. Then the next question, which of the following processes are involved in a horizontal gene transfer? The horizontal gene transfer means gene transfer in a one generation. The conjugation, transduction, and a transformation, these three processes are a processes for a gene transfer, or which are a processes for a recombination, and these are horizontal gene transfer processes. In a conjugation, the gene transferred from a donor to a recipient cell through a physical contact. In a transduction, the gene transfer get via a use of bacteriophages. In a transformation, cell-free or a naked DNA get accepted by a recipient cell. Conjugation, transduction, and transformation, point first, second, and fourth, these three processes are involved in a horizontal gene transfer processes. So option D is the answer of this question. Then the next question, match the class of antibody to appropriate descriptions or their functions. The IgA, it is a secretory antibody present in a body secretions like tears, saliva, mucus and in a milk. The from right column, option P is a matching of IgA, then IgG. The IgG is a most abundant antibody in the blood. IgD found on a B cell membrane. IgE, it gives immunity to the parasitic infection. Such combination is in option C. Here in this image, 5 types of antibody and their characteristics are listed. IgA, this is a secretory antibody secreted into a mucous saliva tears colostrum, and it tags for a pathogen for their destruction. IgD, it is present on a B cell receptor and it stimulates the release of IgM. IgE, it binds to a mast cell and basophils. It secreted or it is synthesized at time of allergy and anti-parasitic activity. IgG binds to phagocytes. IgG are a main blood antibody for a secondary response, and this is abundant in a serum. It crosses a placenta. Then IgM, the IgM is antibody which is produced in a primary response. It fixes a complement and it is present in a B cell receptor. So this is about these antibodies. Option C is the answer of this question. Then the next question, match the diseases with an appropriate causative agent. Rheumatic heart disease, it is caused by the organism Candida albigans, not it is caused by a group A streptococcus. Syphilis, it is caused by a Treponema pallidum, it is a sexually transmitted disease or a STD. Treponema pallidum forms a cankri on a male sex organ, and this Treponema pallidum is one spirochete. Diagnosis of syphilis can be done by using a Fediarel test. Genital warts is caused by a virus, that is a human papilloma virus. Oral thrush, the causative agent for oral thrush is one fungi, that is a Candida albigans, and this causes lowering the pH at site of infection. So first Q, second S, third R and fourth P. Option B is the answer of this question. So thank you for watching my video. Please subscribe my channel Easy Life Science, like my video, share it to your friends, and don't forget to press the bell icon so you will get the notification of my newly uploaded video. Thank you once again.